thanks for joining the third of our the third and final webinar of our webinar series for antibody discovery. Today, I'm joined with two wonderful guests from Twist and Genovac. We'll introduce them in a minute. But before we get to that, my name is Vincent Pai. If you haven't seen me already in the first webinar of the series, as well as part of the Q&A in the second webinar series, definitely head over to Bruker Cellular Analysis and check that out. We have some great case studies and conversations with folks from Twist as well as Genovac, presenting on how they leverage the Beacon technology, as well as their own wonderful expertise for antibody discovery to achieve some great results with their client projects. So yeah, head over to our website to take a look at that. But yeah, today it's a really great privilege and honor to be able to speak with these two folks here. Colby, why don't you introduce yourself real quick and tell everyone kind of what you do.
Sure, thanks, Vincent. So I'm Colby Souders, CSO of the Biopharma division at Twist. So I sit in the Boston office, but work very closely with both Boston and San Francisco sites that we have where we do antibody discovery. So we do antibody discovery both in vitro and in vivo platforms, but as you might have been able to tell from an in vivo perspective, we really enjoy using the Beacon as our core single B cell technology for screening on our in vivo platforms. And so we apply that to a number of different species. We have our own hyperimmune mouse models. We use humanized mice, we use rabbits, we use immunized alpacas. And so we've been able to really leverage the Beacon in a number of different ways to screen those different animals on the system and leverage our favorite feature of it, which goes back to when we bought our first one in 2019, which is the fact that you can do multiple sequential screens. So we really like the depth of screening that we can do and the number of different assays that we can run on the system, and enjoy using it for a lot of our partner projects.
Awesome. And we're also joined here today with Brian from Genovac. Brian, why don't you also introduce yourself?
Hi, everyone. Thank you, Vincent. I'm Brian Walters. I serve as the CEO of Genovac. We're a Fargo, North Dakota-based CRO CMO with facilities in Fargo, where we're headquartered, and Freiburg, Germany, so where we do antibody discovery and production, but manufacture any biologics, plasmid, variety of recombinant proteins. And like Colby, we're big fans of the Beacon instrument and focused on genetic immunization, which our company was founded on, so we can address very challenging targets with a unique combination of immunization and screening vectors we've developed over time, but found the Beacon to be best of class for sensitivity and work with multiple species. So we would do our work in both wild type and transgenic mice, rats, work with rabbits, llamas, transgenic llama mice. We've screened human cells, and on our radar in roadmap are work with organoids and chickens. So we specialize in multiple species offerings and love, again, the sequential assay and sensitivity capabilities of the Beacon instrument. And it's a pleasure to join you all today.
Awesome. Well, thanks so much. Yeah, and for folks online, feel free to submit questions in the Q&A session or section. We'll be reviewing those and kind of sprinkling those in throughout our conversation. We don't have to necessarily wait until the end as we have our conversation. But we'll keep this kind of light, keep this a little informal, and we'll just get to know Colby and Brian and what they think about antibody discovery and where we're headed. Yeah, so I'll dive right in. We back in, I think 2021 was kind of the last time that we had a similar kind of conversation like this, a roundtable. And so kind of since then, I think the world's seen a lot happen. And so, yeah, why don't you, maybe Brian, if you want to start us off, what are some of the major trends that you're seeing in antibody discovery and maybe even specifically a therapeutic development that you're really interested in or gets you really excited?
Yeah, thanks, Vincent. And for us, I think, and a comment we made a few years ago that, I think we continue to see better tool sets from which we can work on our trade and the availability, enhancement, development of single cell technologies, especially the Beacon, again, critical in what we do. But of course, more recently, maybe AI and machine learning tools that we can use for antigen design, do better recovery of B cells once we are able to elicit that immune response and down select antibodies of interest more efficiently, faster, and for less money. So we're focused on more robust discovery campaigns and doing that work faster, saving downstream money, and the tool sets have changed dramatically, even from last time that we talked, in terms of our ability to do that. Our genetic immunization platform lends itself really well to the use of these tools like AlphaFold and other AI machine learning tools, so we can improve antigen design. And again, love the fact that we can work multiple species on the same project, same target. I think another thing we love about the Beacon is we can load different organs from different animals and species, strains in a single run, and then hit it in a second day, going really heavy on where we see the best concentration of hits from whatever strain or species. So love the tool sets that have changed. The work in species has expanded dramatically, as I mentioned in my introductory comments. And then we've been focusing on rapid expression purification of antibodies so that we can characterize and develop them more quickly. Certainly in terms of modalities, a lot of excitement, I think, in use of antibodies, including ADCs, and continued interest in multispecifics, bispecifics, monospecifics, CAR-T, and our ability to, I think, have success against challenging targets has just been enhanced as well.
Yeah, there's a lot going on. Certainly from our side, a lot of different species enabling a lot of folks on some different, host species beyond just our traditional mouse and even human models. I think that really helps with that starting diversity that we start to screen, right, and that gets us the most shots on goal for that potential therapeutic needle in the haystack. Colby, how about yourself? Kind of some major trends that you're super excited about.
I would just start by also just echoing that too, that the different species are really helpful, and like Brian said, just the ability to tailor for each one to maximize. We've done things like optimizing cytokine cocktails for alpaca IgG2-3 secretion to maximize that, and so just having that time and the ability to screen on the Beacon and use that as the tool, not just for the antibody discovery, but for optimizing some of those techniques, is just an amazing ability. And so we've really relied heavily on that, not just as a tool to find antibodies, but a tool for screening methods. And that, media cocktail is one such example of that, but there are a number of different ones too. And I think that plays very well into what you were both talking about with optimizing for each different animal. I think one thing that I would point out is that number one, from a screening tool perspective, I'm surprised at how little the field has changed and challenged the Beacon, to be honest with you, in the last few years. There isn't another instrument out there that can screen in the same way and do those multiple sequential screens and flush the channel above, and all of the great things that we love about that. I guess what I anticipated, to be honest with you, was in the last 3-4 years that similar systems like that would come out with maybe higher throughput or different capabilities, orthogonal capabilities that might enable that. But there really are very few options out there that challenge it in that same way. So I guess that's one surprise that I've seen since 2021. Another one is I know there's been a lot of talk about more difficult targets coming through, but I think one of the striking things that I've personally seen is not necessarily new targets and more difficult targets, but new modalities being used for the same targets. So we do see the same targets over and over again. There's still, multi-pass and really difficult targets have not become the norm yet. It's still, your single pass transmembrane proteins, things like that are the vast majority of projects, but people are using those in new ways. So people are constantly looking to find bio-betters to the same target because they want to target, let's say, a new epitope, or maybe they want to target in a pH-sensitive manner, or they want to apply it as a bispecific, and they need, let's say, a biparatopic that have two very particular epitopes. So I think that's where, again, I really think the screening is important, because we can go after the same target, but you're going to need to find a binder in a different way than you did 3 or 4 years ago, because people are looking to use them in a different way. And so that's where we've put a lot of effort as well to making sure that we can screen in those ways, do really efficient competition assays on the beacon, utilize things like your affinity ranking capabilities that we now have and utilize for assays to get that screening data up front. Use internalization assays to be able to say, well, this set of candidates, these ones internalize faster, these ones internalize slower. And so then that allows you to start to segregate some of those hits at the single B cell level to choose which ones might have that, that might be more amenable to that downstream reformatting as a very different modality than what somebody used that same target for 3 or 4 years ago.
Yeah, I think, Colby, that's a wonderful answer. And actually, you kind of predicted the question in the first question that we got in the Q&A, really around the shift of what antibodies are being used for, and really the different kinds of modalities, and how does that change kind of the services, right? It's really, I think, like you're mentioning, is the different kind of assays. We all thought that, we're just going to get a lot of new targets, really difficult GPCR targets. We're not seeing that, I guess, as, different, or folks are looking at, the risk of taking on a completely new target versus, taking something that may have maybe a safer profile and just repackaging that in a new modality for maybe potentially different indication, but kind of potentially reduces risk. But I think it's been wonderful to see that the system itself, the beacon, is able to kind of pivot, even though we've been focusing a lot on those difficult targets. We're still able to contribute and retain kind of our position there of being able to find really different antibodies than maybe you found before.
Right. Yeah, I guess just one thing to elaborate maybe a bit more for the question in the chat is, we take a peek in at these metadata analysis every once in a while and see what the landscape looks like. And we did that recently and was actually surprised to see that the split of targets and what people are going after from a target perspective has not changed very much in the last 5 years, frankly. It's about the same. It's 50-50 soluble versus membrane protein targets. Of the membrane protein targets, about a quarter of those are your challenging multi-passes, and the other 3/4 are single pass. So interesting to see that, but certainly the end use modality is different. Everybody wants to reformat as a bispecific in the end, or want to reformat as an ADC. And so they're not being used as a traditional mAb in that sense as often anymore, or they're looking for maybe a new blocking characteristic or agonist activity. So very interesting to see that, and repurposing old targets, I guess you could say.
I'd add Colby's comments that the multimodality thing, I think we're seeing more often versus somebody more specifically just looking at bispecific. So they might be looking at monospecific, bispecific, ADC, and CAR-T. So I think for us, just not much of a shift in terms of what we're doing for services. Maybe a little bit that we're working to always create a bigger, more diverse pool of antibodies, and to be able to screen them and characterize them very quickly and cost effectively. So adding small scale expression purification, again, CAR-T instrument, which we can rapidly develop affinity or do epitope bidding in. And so just pushing costs, lowering costs, doing it faster, working with larger antibody sets, which I think also position us well for the people who generate tons of sequences that have wet lab problems in terms of generating data and characterizing those antibodies from those sequences.
Yeah, absolutely.
Maybe one other thing that we're doing a little bit more that I guess we haven't seen is on the challenging target category. So we're doing, I think, because of the tool sets that and the enhancements, especially the reach we get on challenging targets, we're doing more work in the small molecule area against some really challenging targets where they need tools to further their work. And we're doing a little bit more in the diagnostic area as well with co-immunizing rabbits, for example, and multiplex screening, which works really well on the Beacon instrument. So we can now, I think, bring solutions that are affordable for markets that historically wouldn't pay what a therapeutic company might pay for a development campaign.
Yeah, that's a really good point. That's something that I think, again, to your original question, Vincent, if you think about it 4 years ago, that certainly wasn't something, at least on my radar, on our radar, of, we can leverage the throughput and capability, the screening capabilities of the Beacon to screen multiple targets at the same time. And really, like you're saying, Brian, maximize a run for multiple targets. And I think that's one thing that the landscape kind of opened our eyes up to, which are saying, oh, all these folks, they don't need an antibody to one target anymore. They need it to 2 or 3 targets at the same time. And so how do we make the most of that? And you're exactly right, though, that the Beacon enables you to do that, not just to interrogate a single target really deeply, but you could interrogate multiple targets simultaneously, semi-deeply, I guess you could call it.
Mm-hmm. Yeah, there's a little bit of a compromise there in terms of our throughput, but it is really straightforward to just run another assay or leverage a different color on our fluorescent set. And so yeah, and I'll say that even we're doing that internally. Our job isn't necessarily to run client projects like you all, but we want to make sure that we're running a lot of these workflows to ensure you all have a robust product to work with. And so just in terms of kind of some of the economic headwinds we've been facing and macroeconomic things, we've had to cut review our costs and figure out ways to cut that, but still maintain the level of quality that we deliver. And so one of the things that we turned to was immunizing rabbits and mice with this, with 3-5 different antigens and screening against all of those. And that's just been, we don't blink blink when it comes to that. It's just another target, another set of assays. So great question came in. So we'll kind of address this one. Do you think there's a lack of new targets coming through because we aren't able to identify or predict relevant epitopes? Brian, why don't you start with that?
Yeah, I think the tool sets are getting better to allow the identification of relevant epitopes, and we're working on campaigns like that. And again, the genetic immunization construct platform lends itself really well to adoption in some hybrid approaches to target specific epitopes. So we're always exploring new ways to reach those, but we're also seeing a few new targets ourselves. For sure, we're looking at some past targets where we can make better molecules, but we're also seeing new targets as well, especially in the neurological and autoimmunity space. So, and I think it's actually driven by in part by the ability to identify these very specific epitopes that people want to target, and as well as reach, I think, some targets that historically that we haven't had success on.
Yeah. How about yourself, Colby?
Yeah, I think we're seeing it's about the clip of 2/3, 1/3, 2/3 of a similar same target versus 1/3 of new targets. So like you're saying, Brian, I agree. We still see plenty of new targets, a lot in the neurospace, the transporter space, people going after some new transporters, again, with the idea of a modality shift that you can do there with novel transporters. I don't think it's necessarily identifying or predicting new epitopes. I just think there are some targets that are really good targets for particular modalities and indications. It's just we know that there are biobetters out there. We all know that a particular therapeutic it goes down that path for multiple reasons, and not all of them are because it's maybe the best epitope or the best therapeutic. Maybe there's business decisions that are weighing on that. There's time. Maybe it was the first one made that worked really well, but that doesn't mean there aren't better epitopes. And so, yeah, again, to Brian's point, that's why we have different techniques at Twist. We've got the proprietary hyperimmune autoimmune mice that we use to develop really broad epitopic diversity. So if you need to find a hit to an unknown epitope because you don't know what it is, but this is a really good target for your particular indication, Well, it's good to find all the opportunities and then interrogate those downstream to find what that best epitope is. Maybe when the first therapeutic for that target was developed 7, 8, 9, 10 years ago, methods like that were not used. And you had very limited epitopic coverage, let's say. And so you found a hit to an immunodominant epitope, and it worked because it's a really good target, but maybe you can have a bio better. And so I think a lot of folks are thinking about it in that sense. And so it's not because we're not identifying or predicting the relevant epitope, it's just the tools weren't available to do that 5 or 10 years ago when a good target was first identified as indication.
Yeah, yeah, those are really great. Yeah, we're definitely also seeing that relative to people inquiring about, hey, can your beacon do this? And are these the, appropriate targets that that we can enable? One of the really fascinating case studies that that I really enjoy is we have some folks over at the NIH using the beacon as well, specifically for infectious disease, and their goal is actually is new epitope discovery, but they leverage antibody discovery for that. And so they're, again, leveraging that multiplex assay to look for broadly neutralizing antibodies. In and of itself, those broadly neutralizing antibodies could be really nice or really interesting therapeutics to help with disease later, but they're taking that a step further to identify potentially conserved epitopes that already are functional, and so they're using this as a means to kind of just go a little bit beyond kind of your traditional three-dimensional structure, protein structure analysis and your conserved epitope analysis, but going straight to which ones are functional, which ones are relevant. So yeah, I think to Colby, your point, right? It's the tools that we have now, and kind of the understanding of these different antigens have really changed in the last 5-10 years, and so we're doing a lot more creative things there. So even if it's not necessarily new targets, it's new ways to be more effective and more efficacious in the end.
Yep.
Yeah, I think I hinted a little bit about this, but and you all have brought this up, but I think there's a little bit of focus on AI more recently, and so definitely want to hear your thoughts there. Yeah, and so like, where do you see, or how do you use AI today in your all's work, and kind of where do you see this going? There's a lot of talk about de novo, and are we going to be out of jobs soon if this is all done in silico? So yeah, definitely want to hear your thoughts in this space. Maybe we'll start with Colby this time.
Okay, yeah. So in terms of the first part of how we leverage it, so leverage it very deeply on our in vitro projects. Obviously, that's not the focus of this discussion, but in terms of the in vivo projects, obviously it's a big part of sequence analysis and the sequence analysis pipeline when you have a lot, particularly things like when you NGS the full population. So you can derive a lot of value from hit expansion workflows where you have a nice set of well-characterized single B cell hits from the beacon, and then you look at the full NGS population because there's millions and millions and millions of other B cells that you can't screen regardless of the instrument or the technique. And so you can use those two pieces of information together with AI and machine learning to then do your hit expansion workflows. But in terms of the second part of your question, which I think is very interesting in seeing the landscape continuously evolving, is that we're not out of a job yet. Frankly, I hope, well, we won't be out of a job in 5 years, but we'll evolve our job in 5 years because it does work. Because right now we see great traction and the ability of AI and ML to be able to optimize candidates or maybe approach them in individual target-specific ways. But generalizable models have not been developed yet. However, I really hope tools like the Beacon and single B-cell screening can lead to some of those generalizable tools and will put us out of a job, so to speak, or change the way that we need to do our job, is what I would say. So it's interesting because we've seen a lot of AI ML companies leverage two ends of the spectrum. So one is, let's make and test hundreds or thousands of candidates with recombinant material. That works really well to train these models. Then on the other end, it's let's make these very large display libraries. So, 1E7, 1E9, 1E10, libraries and screen those to train the models. Now it's interesting because the beacon sits right in the middle of that from a screening perspective, tens to hundreds of thousands of cells. And it interrogates a different population. Usually we're using an immune cell population. Now, that doesn't mean we can't leverage it for other types of screens. And that, I think, is a very interesting avenue to approach in the future. But we don't see a lot of companies leveraging that ability yet to train models. But the beacon is really primed for that, because not only can you find the positive hits and screen in multiple ways to find different classes of positive hits, let's say, but you can also export and sequence all the negatives as well. And that provides a really rich data set within a matter of a couple of days that you could use to leverage those an immune model for a target specific model. And then again, hopefully generalize that out to a more generalizable model, because like we talked about before, you can turn over multiple targets on the same beacon run, and you can do that all within a day. So the concept of doing 30, 40, 50, 60 targets on the beacon within a reasonable time frame is not unheard of.
Like Colby, loved the beacon and the role it plays in full repertoire screening and the use of NGS and hit expansion, and using AI machine learning tools to help us down select and identify the most interesting candidates to proceed into wet lab production and characterization. And increasingly, I think maybe the focus that we've had in AI machine learning in the short term has been the sort of the light use of engineering tools for humanization and some affinity prediction, but we're getting deeper and deeper using our wet lab to try to solve bottlenecks as well. So both for the discovery campaigns and the abundance of sequences that we can generate, but also if somebody comes to us with a set of sequences that we can cost effectively produce and characterize and develop data. And to Colby's point, I think we're also equally interested in negative data sets combined with the hits that we have to further develop the ability to predict function and affinity and deepen the use of our engineering tools.
Yeah, I mean, one of the things that, Brian, you were telling us about, I think, is a little bit around kind of solving that data bottleneck, right? Off the beacon or off of a lot of these other screening display technologies, you end up with potentially a lot of hits or a lot of sequences to kind of go through. And Colby, you kind of at Twist have a little bit of a luxury to be able to synthesize those internally at the biggest scale in the world, right? And so you kind of have that, but kind of the rest of us, right? We have to kind of develop new tools to kind of break that bottleneck. And so, like, Brian, do you want to tell us a little bit about kind of how you're at Genovac, what you're thinking of to try to get to better characterizations necessarily off of the beacon?
Yeah, so like in present state, we clone into expression and sequencing vectors, and we can really rapidly generate recombinant material, and as well as sequence the antibodies that we're generating. I think long term, I think we've progressed to scaling into, from dozens of antibodies, maybe multiple dozens to hundreds, and I think we want to trend toward thousands to time and cost effectively do that. So we're looking at various synthesis alternatives to be able to do that, and maybe utilize the beacon actually in screening in a different way in a future state. So we're not there yet, but we're investing heavily in that area. And then I would add to, and I think I previously mentioned that we also see AI machine learning, I think, critical to antigen design as well.
Mm-hmm. And then, yeah, definitely don't want to kind of leave you out of this conversation, Colby. But I guess, how do you, Colby, see kind of the DNA printing aspect, or that side of it? How do you use that to kind of enhance this kind of downstream kind of leveraging AI in your lead optimization?
Yeah, so like you pointed out, we have the nice luxury of being able to do this at the scale of hundreds to thousands, to 10,000 or more candidates, and so we can do it really quickly. And so that's one of the main concepts that we've seen folks focus on, is how fast can you turn the data over, because we need to train the models and then do another round. And the most valuable models have come out of those multi-round training methods. And so to be able to turn over going from a sequence to having purified protein for screening in about 10 to 15 days is a huge advantage for these companies, because then that reduces their cycle time by quite a bit. And I think that's one of the keys, is not just the data quality and having the breadth of data, but not taking 6 months to a year to turn around a 2 or 3 cycle, but being able to do that in a matter of a month or two. If we take that one step further and think about what this could be envisioned in the future, it'd be nice to have something like the beacon as the center of a closed loop cycle too, because there's a lot of other instrumentation that we could put around that and automate to make sure that we have cells coming off the beacon, maybe into an automated sequencing platform, maybe onto automated synthesis, production, characterization, and then that can feed back in. And so, I think it's a nice home in Bruker to have the beacon in, because there's a lot of other instrumentation and capabilities from parent Bruker that may be able to integrate in very nicely in a closed loop cycle like that.
Yeah, definitely excited. We've already started conversations with a lot of our sister divisions. There's a lot of excitement in building out essentially that ecosystem that we're envisioning, kind of maybe not necessarily all the machines on a conveyor belt, all fully automated, but that's the dream, right? Is maybe we can kind of get towards that. We have another question in the chat, or at least the Q&A. Just with the market conditions that you're seeing, how does that look in terms of projects that you see outsourced to service providers like you all versus kind of what we would like to see is definitely in-house running a lot of these. How have you seen that shift, especially in the past couple of years, and where you see that kind of trajectory going this year? And do you see AI potentially consolidating some of those services? We'll start with Colby.
Sure. Yeah. So I don't think it is consolidated. It's certainly not consolidated in the near term. Quite the opposite, has expanded, because again, a lot of these models need to be trained very heavily on reliable data. And so I think that has led to an expansion of the outsourcing and use of these types of techniques and the things that we've been discussing. I don't see that changing in the near term. Because once again, I think folks are going to find that they can get a good model, but then if you continue to train it, you get an even better model and a better model. And when that eventually that will plateau. But I think also the capabilities of those models and the scientists running those models is going to continuously improve, and we're going to learn new things. So it's just like I'll make the analogy of what we were talking about earlier with different targets. Same target, different modality. So it'll be the same thing here. Same AI, different use cases, and we'll need to retrain data for those use cases. And so I think it's been a great boon to the industry and really helped expand different use cases beyond just therapeutic discovery, now onto training new platforms and systems to do future therapeutic discovery. And I don't see that changing in the near term.
Brian?
I would completely agree with Colby's position and what he's seeing and what he foresees in consolidation. And in terms of number of campaigns being outsourced, I think to some extent historically it's been cyclical with some of the larger companies. They kind of expand and contract. And for sure, I think there's been a lot of concentration on preserving capital for things that are in the clinic in recent years, and a lot of downsizing of preclinical and discovery teams. So I think we've seen a little bit of a trend of that. I would say some being cyclical of more outsourced campaigns, but I would expect as things pick up and new targets are identified that we would see a trend of more outsourced campaigns. And then I think there's also an impact of where the dust settles in terms of relations with China and services that are being performed there, and some things like the Biosecure Act and maybe some risks associated with development of programs in China. I think that could very well influence outsourced campaigns and in the United States and Europe, I think very positively.
Definitely.
I would just add too, we've also seen a nice blossoming of smaller companies now, whereas, let's say 2 or 3 years ago, when we had the biotech downturn in full force, companies were certainly sheltering cash and we're in preservation mode. But I think we're firmly seeing people break out of that shell and want to take advantage of, again, these new modalities, new ways of going about it, and don't want to be left behind as other companies are doing that. So I think we see a nice acceleration of that new discovery and risk taking that was not there a few years ago.
Vincent, if I may add to that maybe one other dynamic I think that is leading to a little bit more outsourcing. Some of the new companies that are being funded and formed, I think have learned lessons from the past and how they use their capital, and they're electing not to buy expensive instruments that they might use for one target, operating a little bit more virtually, and we've seen that trend and dynamic and a little pickup in business from companies I think that historically would have been tempted to open their own labs and again, when you add capital and space and headcount and where do you want to use your resources and your capital, and I think they're choosing to add more value downstream than in the discovery area.
Yeah, certainly. We have a question in the chat. Kind of what's the cost per cell on the Beacon, and are there plans to increase the scale to screen millions of cells per experiment? I'll start since I think this is more Beacon related, but definitely Brian and Colby can let you all chime in there. Cost per cell, I think it really does depend on the type of campaign that you're looking for, kind of throughput that you're looking at. Are you bringing your own reagents, your own antigens? What types of assays you're doing? One of the really nice things about the Beacon is again, like folks have been mentioning, is lots of assays is really the key here, is being able to run and get a really deep understanding on every B cell. And so while I think the upfront cost, if you're doing something really simple, right, a soluble target, maybe you're just looking for binders, might not be the best economic fit for our technology. Doing something like looking at its particular epitope or its function, or looking at different kinds of binders across different antigen types, whether it's just a linear transcript, or it's recombinant, or if it's on the membrane of a cell. I think the more you kind of stack into the technology, the cost it's not just necessarily a cost per cell calculation anymore. It's kind of cost per characterization that you get upfront that you don't have to do downstream later. And so that's at least the way that we see the Beacon and how we kind of justify the somewhat premium cost to that. Maybe this will segue into the next topic, but just really quick, relative to increasing scale. We don't have, we get this question a lot. We don't have immediate plans to kind of increase that scale, but certainly that is on our roadmap. It is something that we are thinking about and evaluating new technologies, ways to innovate both our consumables, definitely, but then also maybe even a platform to kind of increase that throughput, that screening throughput, and really trying to bring as much data to the customer and maintain that depth, or even expand that depth that we've been able to provide. Maybe Brian, I don't know if you want to comment on kind of the from the service side, what you all see.
Yeah, in terms of the throughput in screening more cells, I think Colby mentioned repertoire screening and doing NGS work and hit expansion. We have a second approach to that as well. That we have a gravity platform that we'll use to screen excess cells. So we'll do the primary screen, run sequential assays on the Beacon, but saves a little bit of time to go identify binders that we can then very rapidly again express and purify and characterize. So we have two methods to increase the scale. One is the hit expansion workflow that Colby described, and the other is to load cells onto the platform. We can do 1 million cell screens, but doesn't have the ability, the elegance that the Beacon has to do sequential assays.
Colby, anything to add?
Yeah, I would say from our perspective, from the cost perspective, that's why we have intentionally built out multiple services. So like Brian's saying, there are alternative strategies. Our alternative strategies are still to make sure we have a hybridoma platform that's very robust, and then also an in vitro screening platform, because for any given target, it's not necessarily the throughput that matters or the screening capabilities, but maybe you need a specific property from that target. Maybe you have a very specific TPP profile that you need. And so one of those different discovery methods is more well suited to meet that TPP criteria. We feel the Beacon has a very wide dynamic range within that, so to speak, and so it fits most of those. But certainly from cost perspective, throughput perspective, particular TPP profile, you can leverage those different capabilities. Also, from the scale side, I would touch on something Brian said too, which is we can, let's say, use intentional scale, meaning you get a lot of information from the initial screen you run on the Beacon, and then the next day you could screen from the best performing cohort or immunized animal, mouse, what have you. Then you have other animals perhaps in the same immunization scheme, and then you can go back to more of those. And so it's not, again, it's that concept of sequential screening, not everything in bulk in massive scale all at once. Let's learn from the data we get from round one to have a more intentional round two. So screening 20,000 cells in round two with an informed data set is more powerful than screening 2 million cells in round one sometimes. And so we certainly like to leverage that approach and take that of let's learn from the screen, let's learn from what worked. And because of the speed of the Beacon and the single B cell screening output that we get, we can do that really quickly. Yeah, it's a 1 day process. So then if we learn something on day one, day two, we can immediately apply those learnings. And it's not a multi-month, not even a multi-week cycle. It's just a couple of days.
Yeah, no, that's definitely. Thanks for bringing that up. We love that aspect. Even here is, we're always kind of working with our typical model systems, but whenever we get that one-off, hey, can we do this particular target? And it's the first time we've ever worked with it. Yeah, we do that exact approach. Is let's try to learn as much. It's not in that first run, it's not necessarily that we're going to deliver the best antibody that very first time, but it's about that rapid learning cycle that we're able to do in that single day workflow. We run as many assays as we can. Sometimes we don't export, but we sit down, we look at those results. We say, hey, which assays made sense? Which ones didn't? And where are we seeing, and how should we actually adjust the types of assays, the types of chips, the particular animals or the wells that we're working with for activated cells, and then we really go after the second round, and that we get really, really great results from. So yeah, I definitely think that there are ways around necessarily just more may not always be the right approach.
Right.
Yeah, but I think now that we have our kind of juices talking about kind of future state, I know you both of you have been working with us for a number of years now, but giving you the first chance to publicly ask, hey, if you had the opportunity, what would you want to see out of BCA? What new features or what new capabilities would you like us to be developing, or actually just more broadly, where would you like to see the market or other innovative companies kind of push and develop so that we can really all together kind of push antibody discovery? Brian, why don't you start with this one?
Yeah. As stated by Colby and that I share, we're very happy with the platform. Love the work that you've done to expand species, and we partnered in that area a couple of times, and more things that information we can get faster from our screening, including the affinity assay, the affinity ranking assay, which we really like, and continue to see that feed our prediction capabilities. And I think maybe more, you touched on cells. Of course, we always want to screen more cells, have more exports, but we have regular discussions on that, and love the sequential assay capability, more functional assay work, and I know we're collaborating in that area as well. And so I think just anything that helps us identify really the most interesting candidates faster for less money is what we're focused on as a company, and maybe specifically to the roadmap and that you guys have. I think anything that we generate a lot of data from the Beacon instrument. I think anything that can help us grab value from that in a streamlined way, and in terms of the files that we're generating and analyze and use that to improve assays. And I think it'd be something that our team would mention, or probably has mentioned to you, Vincent.
Yeah, definitely. Colby?
Yeah, I just want to touch too on the affinity assay and kind of some backstory on that, and that realizing that you could do intensity measurements on the Beacon is something that was, we knew that was capable 5-6 years ago. And so we were talking to you about that, and how do we leverage that? And so it's great to see that you were able to take that in, develop the software updates to be able to then leverage that feature that was inherently there. So I think that's a great success story there, and an excellent feature on the Beacon to be able to have that affinity ranking and take advantage of something that was there in the hardware and capabilities, and then do software updates to be able to deploy that. So really enjoyed that as a success case. Now I think moving forward, won't touch on it again in depth, but again, combining with multiple instruments, lab in the loop concept. I know that's probably a bit further out and much more complex to do, but in the most basic iteration, you could think of pairing it with a sequencer, so that then that's a fully automated process, not just from export, from cells on to export off, but also all the way through to sequence delivery at the end of it. So more of a fully automated package would be excellent. Just to touch on another hardware thing, more optical trains. So being able to simultaneously interrogate multiple chips at the same time, just to save time, basically, right? So then the screening time is compressed, so then we can screen more chips in less time. And so that's an indirect way of handling that. How do we increase the throughput? I'm not an engineer, so I have no idea how hard it is to stick a second or a third optical train in that box, but I suspect it's not easy. And then from an assay perspective, I think going beyond just saying, well, screening B cells, what other types of things can we screen? Can we screen other types of cells or non-cell components? Would be a nice way to expand use cases on the Beacon. And finally, from an assay perspective, more developability assays. I think that's becoming more and more of a focus, certainly for AI ML companies, to be able to engineer and optimize algorithms to predict developability profiles. And so the more of those assays that we can integrate onto the Beacon, just the stronger that depth of screening output becomes.
Yeah, I have them all written down, and we have very regular conversations around this. So definitely good to kind of share that with folks of how it is that we're taking that feedback, and we're trying our best to really try to deliver upon those, not just for just to help Colby and Brian, but really to push and really drive antibody discovery success in the market as well. So. We'll have one more question, and I think that we'll conclude just given time. But let me post that before that. But how challenging is it to predict neoantigens from neoplastic cells that interact with BCR sequencing, or by sequencing them using the Beacon? Is that something that you all have done or tried? I'll start. We haven't done that yet, but we're really interested. Neoantigen discoveries keeps coming up every so often. We're a little bit focused here in this conversation right now, and my expertise is really within antibody discovery. But another large application that we have is within the T cell space, where we're looking at looking at TCRs and how those interact with potentially neoantigens from primary tumor cells. And so that definitely comes up in that forum a lot as well. But I think it's still early days that we haven't fully demonstrated that capability, but a lot of really great ideas on how to do that on the Beacon. Colby or Brian, I don't know if you all want to kind of chime in on that as well.
Yeah, I could start, but I think we've had a couple of projects like that. I think some of the keys, if you're going after a completely unknown target, for example, are if you've got a very straightforward functional output, meaning you've got a cell, it's got a reporter, it fluoresces green when it's activated, or doesn't when you block it. And so then you can immunize with some pretty interesting cocktails or whole cells or something where you don't know exactly what you're targeting, but you're looking for that very basic functional readout. And then that can be one fairly straightforward method to use for, let's say, target interrogation. And then, once you get those hits off the beacon, now you can backtrace those to what you were actually targeting on that cell to elicit that functional output. So I think there are a few ways to do that. It's certainly a less popular use case, but I don't think there's a reason why it can't be a more popular use case on the beacon.
Yep.
And we have not used that workflow yet. As Vincent, as you know, we just implemented the T-cell workflow, but we'll certainly take this back to our team and we'll explore it.
Yeah, the only thanks for those. The only thing that I'll add to that is, there's actually a really nice paper from the NIH group that I mentioned earlier, really around discovering a neoantigen for malaria. And there's a really interesting kind of analysis that Joshua Tan over at NIAID kind of uses the beacon for. Again, as I mentioned, it's about neoantigen or neoepitope discovery through antibody discovery. And so if you get a chance, you can definitely check that out in our publications list. It's one of the really exciting recent publications there. I don't think we see that necessarily as frequently within the therapeutic space, but definitely within academia. This is something that people are kind of challenging is, hey, can we leverage the beacon this way? And I think that's a really great early case study as to some of the success that could be done if you didn't have a preconceived notion on what the antigen was, but you knew it was expressed on a particular cell type. Yeah, and so I think that'll conclude today's roundtable. Really wanted to thank Colby and Brian for your time just to have this chat. It's always great to see you all and discuss. I think both Genovac and Twist will be at Festival of Biologics next week. So I think both of you all have booths there. And then also looking forward, PEGS will also be present. I think the three of us will all be there as well. So if you all are there at those conferences, definitely come by at our booth, say hi, ask us more questions, tell us what we want to or what you want to see out of the Beacon or out of Genovac and Twist. But just want to say thank you to you all for your attention and for joining us on this webinar series. We appreciate it.
Thank you, everyone.
Thanks.
Thanks, everyone.
See ya.